Okay, so our last talk of this session is by Evi Mercken, and she's coming to us from Belgium. The title of her talk is A Combination of Metformin and Galantamine Exhibits Synergistic Benefits in the Treatment of Sarcopenia. So thanks very much for joining us.
Thank you. Good afternoon, everybody. I'm excited to be here to show some of our data towards a healthier lifespan. I'm going to shift gears a little bit as the previous speakers talked about brain, and I'm going to focus on muscle. So for those of you that are not familiar with Rejuvenate Biomed, we are a clinical stage biotech company that was founded in 2017 by Dr. Anne Belien. So our main effort focuses on the identification of novel safe drug combinations that would operate synergistically to promote healthy aging. So Rejuvenate Biomed consists of two structural components, the platforms and the identified assets. The platforms are made up of an in silico arm and a warm drug-based screening arm. The combin agents are in silico platform, and here we predict drug combinations that target synergistically complementary pathways related to aging and disease. The drug combinations revealed by our platform will then be validated in C. elegans, in part by using our CellEngage platform, and they will also be validated in preclinical mouse models. Validated drug combinations will then be advanced in clinical trial investigations targeting a specific age-related indication. I will now walk you through our first effort to establish a proof of concept for our approach. In the plot shown here, we identified a combination of galantamine and metformin that would operate synergistically in the treatment of sarcopenia. So since it was our first drug combination, we have termed the compound RGX-01. So most of you are familiar with metformin, and it's used in the treatment of diabetes. And some of you are maybe less familiar with galantamine, but it's a choline esterase inhibitor that is used for the treatment of Alzheimer's disease. Just very briefly, I want to mention that sarcopenia is a muscle disease which affects the lives of over 80 million people worldwide. And it mostly affects the elder population, and at current, no treatments are existing. And then based on our platform prediction, we sought to test the effect of RGX-01 in various preclinical models with the main focus on sarcopenia. So our hypothesis was quite simple, and we hypothesized that a combination of metformin and galantamine would have a synergistic beneficial effect in the treatment of sarcopenia. And to test those beneficial effects of RGX-01, we employed a preclinical worm model and also two mouse models. We used a transgenic mouse model, and we also used an H wild-type mouse model, for which I will come back later. So here are the data shown of RGX-01 in C. elegans. We first looked at the lifespan, and here you can see on the left-hand side we have four groups. The black line represents the untreated worms. The metformin-treated ones are highlighted in blue, and the galantamine-treated animals in orange, and whereas the drug combination RGX-01 is represented in purple. And you can see here that metformin increased the lifespan compared to the untreated animals, as was in line with previous research. Also, a significantly increase in lifespan was observed for the galantamine-treated animals, although to a lesser effect than that was observed for metformin. And the strongest lifespan-induced effects was observed for the combination drug RGX-01, which the effect was greater than the individual compounds. As an activity measure, we measured the mean speed and also the pharyngeal pumping rates, and both the mean speed and the pharyngeal pumping rates were significantly increased in RGX-01-treated worms, and here as well compared to the untreated as the individual compound-treated worms. We also looked at the muscle fiber integrity, and as you can see on the figure on the right-hand side, the RGX-01-treated worms had a better muscle fiber organization relative to the untreated worms. And here the data in the worm, they indicate beneficial effects on the muscle structure and muscle function, and these data were used to pursue further studies in 2 preclinical mouse models. So on one hand, we have the transgenic mouse model, and on the other hand, the aged mouse model, which I will come back to you later. So we have selected the inducible muscle-specific OPA1 knockout mouse model, as it recapitulates in just 3 months all features of sarcopenia, such as muscle atrophy, exercise intolerance, and muscle strength loss. Thus, the strength of this model is the short duration before the onset of sarcopenia. So in our study, 5-month-old male OPA1 knockout mice, they were treated for 12 weeks with the individual compounds metformin and galantamine, as well as the combination drug RGX-01. And for the purpose of this presentation, I will only show you the phenotypic characterization data related to sarcopenia. As in the slide shown, you can see that the muscle gastrocnemius weight and the cross-sectional area of the myofibers was significantly increased in RGX-1 treated animals compared to the untreated and individual compound treated mice. If we look at the functional outcome, we see that RGX-1 treated mice had an improved physical performance as they run 3 times as long as untreated as well as individual compound treated mice. And consistently with the improved physical performance, also the absolute muscle force and the specific muscle force, which is normalized for muscle mass, were significantly increased in RGX-1 treated animals. And here we can conclude that RGX-1 is to improve the beneficial effects on muscle mass and muscle strength and physical performance, you need to have both compounds. So transitioning from the transgenic mouse model to the aged mouse model, here we tested the effect of RGX-1 relative to the controls, where early signs of sarcopenia are emerging and slowly progress. So in our study, we treated 22-month-old male mice for 18 weeks with RGX-1, and here also we monitored physical performance, muscle strength, and muscle mass. And here I will also show you additional data about the potential underlying mechanisms. So here, the data of the physical performance on the left figure, you can see that RGX-1 treated animals, they exhibited an improved physical performance as indicated by a longer running time. In addition, the treated animals also had an improved grip strength compared to the untreated animals, and these beneficial effects on physical performance and muscle strength were independent of an increase in muscle mass of mean fiber size. So these data would indicate that RGX-1 has a more beneficial effect on functional outcome than on muscle mass. And as the physical performance and muscle strength improve without having an effect on muscle mass, we next reasoned that the neuromuscular junction would be affected. And for this, we monitored the neuromuscular junction morphology by staining the postsynaptic acetylcholine receptors in red, and the pre-nerve terminal was stained in green, and we monitored how many fibers of the whole EDL muscle did not show a good overlap between the 2 markers. And as you can see in the images, the RGX-1 treated animals, they showed a better neuromuscular morphology with a good overlap between the pre- and postsynaptic sites. When we quantified these images, we observed 15% less denervated fibers in the RGX-1 treated animals compared to the untreated animals. So next, we measure the effect of RGX-1 on the overall metabolism in aged mice. So the mice were put in metabolic cages, and the mice treated with RGX-1 showed higher values for all metabolic parameters in the light as well as in the dark phase. And this is known to be declined with advanced age in mice and elderly people. And as I showed you before, an increase in physical performance, and here we show an increase in energy expenditure without having an effect on lean mass, we next examined the effect of RGX-1 on the mitochondria. So to determine the structure of the skeletal muscle in more detail, transmission electron microscopy was performed on the EDL muscle. And as shown in the top left figure, the sarcomeres displayed a Z-line misalignment, and also the mitochondria showed an abnormal morphology. They showed a reduced number of crista, an electron pale matrix, and on the right side is swollen mitochondria shown. When we treated the mice with RGX-01, Z-line misalignment was reversed, and also the mitochondria showed a better mitochondrial morphology. We also quantified the damaged mitochondria, and as you can see in the figure, the RGX-01 treated animals reduced the damaged mitochondria by half. And because it's known that mitochondrial dysfunction triggers the expression and the secretion of cytokines such as FGF21 and CDF15 from the skeletal muscle, we next tested those cytokines as a readout of mitochondrial functionality. And as you can see in the figures on the right, both blood FGF21 and urine CDF15 were significantly reduced in the RGX-01 treated animals compared to the untreated, suggestive of an increased mitochondrial activity. It's well established that autophagy impairment is more present in aged mice and elderly people, and that's why we investigated different autophagy markers in the formation and degradation of the autophagolysosomal process. And as a first step, we tested whether RGX-01 would reactivate autophagy by measuring the LC3 and P63 changes. And as you can see on the left figure at the mRNA level, we did observe an increase in the transcript levels of P62 and LC3. And despite that increase at the transcript level, at the protein level, both the non-lipidated and lipidated LC3 were significantly reduced in the RGX-01 treated animals compared to the untreated. And this would indicate an increased degradation as a compensatory mechanism for the transcriptional upregulation, and therefore an autophagy flux restoration. Autophagy impairment is also consequent to a lysosomal failure, and as a lysosomal readout, we monitored the multivesicular bodies, and as you can see here, we confirmed the presence of these structures in the muscle of aged untreated mice. Upon RGX-01 treatment, we did observe that these multivesicular bodies were significantly decreased. And in addition, we also, as these structures are known to be positive for LAMP1, we immunostated the skeletal muscle with LAMP1, and we observed a reduced LAMP1 positive puncta in RGX-01 treated animals compared to the untreated animals. And as autophagy impairment also increases the centronucleated fibers, and autophagy is also an important player in the stem cell maintenance, we first looked at centronucleated fibers as a marker of muscle regeneration. And as you can see in the figures on the left, RGX-01 treated animals had a reduced number of centronucleated fibers compared to the untreated animals. And we next wanted to know whether the reduction in centronucleated fibers would be due to the preservation of the muscle stem cells. And for this, we looked at the transcription factor PAX7, which is a marker of the muscle stem cells. And here on the figure on the right, you can see that the RGX-01 treated animals had an increased percentage of the PAX7 positive cells relative to the untreated animals. And in conclusion, we do see effects of RGX-01 at the neuromuscular junction, at the skeletal muscle, and at the circulation. Specifically at the neuromuscular junction, RGX-01 increased the neuromuscular junction stability. At the skeletal muscle, we observed an enhanced autophagy and lysosomal trafficking, a preserved mitochondrial health, and we also have data to support the stem cell reservoir maintenance. And in data not shown, we also observed a reduced muscular as well as a systemic inflammation as a result of the RGX-01 treatment. And here I also want to emphasize that RGX-01 enhanced muscle strength and endurance by improving more the muscle cell quality than the quantity. And returning to our proof of concept, we have shown the effectiveness of our pipeline to predict the drug combinations that are effective in treating a specific age-related disease. Specifically here, we have shown that a combination of metformin and galantamine, known as RGX-01, can have beneficial effects in the treatment of sarcopenia using preclinical models. We're also currently looking at additional drug combinations that are coming from our in silico platform, Common Age. And we've recently finished a 1B clinical study with RGX-01 with a calcing-induced muscle atrophy model, in which we are finalizing the data right now, and we will have the data in the next months about the primary and secondary outcome. And here I would like to thank, of course, the whole Rejuvenate team, and especially thanks goes to Marco Sandri and Katerina Tess, who performed all the animal experiments. Thank you.
Thanks for that great talk. You have a question?
Thank you very much. I actually have some questions to follow up, but I want to make a much larger point. When the FDA has indications like sarcopenia or frailty, in my mind, it's a tough indication because it's an end stage. And that's why sarcopenia has been unsuccessful. People have shown what you've shown in aging animals, right, in models, but it's not the human sarcopenia. So I think your phase 1 study was well designed just to show misuse of muscle, but it's not the same as sarcopenia. So before your phase 1 study, I just want to make a point to everyone. We want to prevent sarcopenia. We want to prevent frailty. And sometimes those indications are tough, as it's been shown.
Yeah, that is correct. We chose first a muscle disuse experiment because it's short, and we want to know if it has effects before going into a chronic study, because it takes 2 years. So, but that's our next goal. If the data are coming out positive, then we will move to the chronic sarcopenia.
One more?
Yeah, thank you. Was there a bone phenotype in the treatment group with RGX?
I'm sorry?
Was there a bone phenotype?
We are looking at the phenotype at a different study. So we're also doing currently a study with Rafa de Cabo. It's a health span and lifespan where the bones are being analyzed at different ages. But at this point, we don't have the data yet.
One very short last one.
Yeah, it's a very nice talk. I just have some, Yvonne Fan from University of Oslo. I have a comment regarding the autophagy part. You find LC32, which is reduced. Normally, it's increased can be like marker of autophagy. And you also check LAMP1. Normally, LAMP1 increase correlated with lysosome. So you may want to do more experiments before you want to give a conclusion on whether your compound can regulate autophagy or not.
Yeah, to come back on your first comment, at the LC3 we did see an upregulation at the mRNA level. So, and we compensated actually because at the protein level it went down. So it's basically consumed. So we have less LC3 at the protein level, which we can conclude that is an increased degradation, because at the basal level we know we have an impairment, and if you have an increase at the transcriptional level and a decrease at the protein level, this would mean an increased degradation. So the autophagy flux is better.
Thanks very much.
Thank you so much.
Thank you.